Hey guys, it's Charles Beasley and welcome to Good Clinical, a podcast and YouTube channel dedicated to showcasing the future of medicine and human health. Today's episode is another in our partnership with Te Titoki Mataora, and today's guest is Dr. Victor Dieriks. Victor is a neuroscientist studying the early effects of alpha synuclein in Parkinson's disease and multiple system atrophy. Very smart man. Please welcome to the podcast, Dr. Victor Dieriks. Parkinson's disease, I've worked a little bit in my previous life as a clinical project manager for a pharmaceutical company. Always found it very fascinating that there's so much research going on in this arena, and yet we don't have any, say, functional cures or anything that per se really does a good job at slowing the progression. So it's quite an interesting area. A lot of research, but doesn't feel like there's a heck of a lot of progress, for example. So what is your lab set up in this context to research in particular? What's your bread and butter?
So let's start off with the first thing, Parkinson's disease. It's actually a wrong word because in disease means like one cause, one disease causing effect. And for Parkinson's disease, we should actually be calling it diseases because we know there's multiple causes. There's genetic factors. You hear a lot about people having their father and their grandfather having Parkinson's disease. But then there's also like clear links to environmental exposure. So there's pesticides that can cause disease as well. So the problem with Parkinson's disease is that there's a whole range of causes, and they will all result in a sort of a symptom profile that we know as Parkinson's disease. And it's very typical. It's like the slowest of movements, the hunched over, the shaking, very typical on one side. But at the end of the day, Parkinson's disease will affect the entire body. People will lose their sense of smell, which is one of the things that I was focusing on early on during my research. But constipation is another one. REM sleep disorder is also one that kind of preludes Parkinson's disease. And one of the things that we don't really see is dementia, depression, because Parkinson's disease is typically characterized by the motor symptoms, but there's a lot of non-motor symptoms that happen as well. And so what my group and I are focusing on is, as you mentioned, that it's the protein alpha synuclein. So in Parkinson's disease and multiple system atrophy as well, which is like a accelerated form of Parkinson's disease, I call it Parkinson's disease on steroids, basically, because it progresses a lot faster, a much more aggressive disease. So within these diseases, there's one thing in common, and it's alpha synuclein. And so what alpha synuclein does, it enables our neurons to communicate with each other, really essential. But in Parkinson's disease and MSA, multiple system atrophy, these alpha synucleins will start clumping, it will form protein clumps. And the formation of these protein clumps is toxic to the brain. And interestingly, so once you have one protein clump formed, it's basically a template for more clumping to occur. So you have this clump that forms, whatever the cause was, genetic or environmental. It forms more and more clumps. It will then spread from one cell to the next cell, to the next cell, to the next cell. And before you know it, 5, 10, 20 years later, your entire body is full of these alpha synuclein clumps. And you can imagine that that's not a good thing for your body because it disrupts a lot of normal systems. It will kill certain neurons within your brain, and it's the dying off of those neurons that will cause like the typical symptoms. And the standard that they say is that the motor symptoms, so the tremor, will actually start occurring when about 70% of the neurons within a certain sub part of your brain, the substantia nigra, die off. So you can imagine that if 70% of something dies off and you only start seeing like symptoms, then there's a lot of redundancy in the brain, and we can cope with a lot of things. But once that you get to that point, it's a gradual decline and decline because the cells keep dying off more and more. So what we are doing is we're trying to see how we can stop that protein clumping from occurring, and more importantly, how we can stop it from spreading. Because it happens as we age, we get wrinkles, we run a bit slower and things like that. So there's a gradual accumulation of rubbish within our body. We're not really good at cleaning it out completely, but if there's if it were to happen and our body can't clean it out, if we can then contain it to a certain brain region and it can't spread to the other regions, then we can actually delay the onset of the disease by 5, 10, 20 years, or it might not even happen. And that's kind of the core function of my research and that of my group.
So the formation of these clumps, that's quite idiosyncratic of a lot of neurodegenerative diseases, right? So Huntington's disease, they have the accumulation of Huntington. In Alzheimer's, it's beta amyloid and tau, yeah. And then for Parkinson's, it's the accumulation of alpha synuclein. I spoke to another neuroscientist, and she mentioned that perhaps because this kind of schema is conserved across multiple different, diseases, syndromes, however you want to call them, perhaps it's like almost like a adaptive a defensive mechanism. Like why is that response, clumping, why is that conserved across so many different diseases? It seems quite interesting. Do you have any thoughts on that?
So yeah, it's all these neurodegenerative diseases seem to have like that clumping of a different, and it's because it's a particular protein, it affects a different region of the brain, and then it causes a specific symptom profile. So with Alzheimer's, it causes dementia primarily. Motor neuron disease is another one. The actual reason behind it's and why it's specifically those proteins for alpha synuclein is quite commonly expressed. And so in our bodies, at a cellular level, a lot of things go wrong. But for some reason, and this is through evolution, genetic regulation, every time there's something wrong, if a protein is misformed or if it's defective, the cells have mechanisms to correct for it. And within these diseases, there's always this underlying mechanism of it not being cleared, not being resolved. For instance, there's a lot of genetic risk factors in Parkinson's that involve defects in the lysosomes. And the lysosomes are basically our waste disposal units of the cells. So if there's something wrong with sending clumped protein to the lysosomes, it then gets stuck. It can then grow. And so in most cases, there's multiple redundancies. A cell can die completely. It can be eaten by the microglia, which is our immune system. But if multiple things go wrong, then you can get these diseases.
Yeah. So it's like the clumping per se might not be the issue. It's the clearing of those clumps, or the clearance, or the breaking them up, or digesting them, removing the cells that aren't processing them properly. That's the issue, because it's the overtime accumulation, the disruption of normal cellular processes, and then the spread of that over time. So what role does alpha synuclein have in normal cellular functions?
So we know a lot about it, but actually we don't know a lot about it. So one of the early functions that it's been attributed, and it's probably still the most investigated, is that it plays a role in neurons communicating with each other. So synaptic plasticity. So when there's vesicles that are being formed, alpha synuclein will be involved in that. Recently, there's been new roles of alpha synuclein in the guts, potentially interacting with the gut microbiome. Okay. But interestingly, what people seem to forgetting. And so the first why synuclein is called synuclein is because it's was first found in the nucleus, synuclein. But not a lot of people are actually looking at the neuronal, the nuclear function of synuclein. So there's a lot of things that we don't know yet.
Gosh.
So
okay.
Still to be continued, I would say.
So I can imagine that that's quite an interesting thing to study then, because there's so many different potential avenues of investigation of this one little protein.
Yeah, and it's when you look at the protein itself. So we have this one little protein, and we used to think we have our gene. A gene forms RNA, that forms a protein. That's how we all got taught at uni.
Central dogma.
Central dogma. Now, that's no longer the case. It's a lot more complicated. So one gene can result in multiple different proteins. They can be shorter. They can be post-translationally modified, so we can add stuff onto it that changes their function. And in the case of Parkinson's disease, really interestingly, they can clump together, right? And so one of the things that I haven't mentioned is what we found is that in Parkinson's disease, these clumps will take a certain 3D structure. And you can compare them very easily to different forms of pasta. Everybody likes pasta, but pasta comes in like 100 different varieties. You've got macaroni, fettuccine, spaghetti, all exactly the same ingredients, but the shape is quite different. And so my kids, they like one form of spaghetti, but they don't like the other one because it tastes differently, even though ingredient-wise it's the same thing. So for alpha synuclein, these clumps can be formed in different forms. They can be a spaghetti or they can be a fettuccine. And so what we found, right, is that how this forms will actually result in a different subtype of the disease. So Parkinson's disease has one, we call strain of a synuclein, one type of pasta, whereas MSA, the faster, more aggressive one, has got a different one. And so interestingly, when we take a piece of brain of somebody with Parkinson or multiple system atrophy, we mush up the brain, we put it in rats, we wait a few months, we'll see either a Parkinson phenotype or a multiple system atrophy phenotype. And we can do that again. We can take that rat's brain, we can mush up a bit, do it in the next rodent, and we'll get the same disease. So it's kind of like a prion, in that it can propagate, but not completely, because it's not that you're gonna, you actually have to have this protein clump within the brain, and it has to be able to spread there. It's not like if you eat it, you're gonna get Parkinson's disease. So,
are there just two main types of these synuclein strains?
There's probably more. But there's, so when we look at Parkinson's disease versus multiple system atrophy, that's like where we see a very clear difference. Within Parkinson's disease, there's probably multiple. And one of the things that we're doing in my group is we're trying to see if the type of form/strains, if that's linked to a certain subtype of the disease. Because that's where the proof is in the pudding. Is if we can identify what kind of strain you have, we can adjust the treatment specifically to that strain before the person will develop a certain profile of symptoms. So we can preempt it.
And that would be also inform their prognosis, right? If it's faster progressing, slower progressing, like you mentioned, treatment interventions, lifestyle interventions, that kind of thing. Well, why don't we pivot to the available treatments that are out there right now? Because again, I kind of mentioned earlier that most of them that are out there, at least pharmaceutical interventions, they are purely for symptoms, right? They're not stopping the buildup of clumps. They're not stopping the dying off of the substantia nigra or other parts of the patient. So, two questions, I guess, in there. What's out there right now, and where does it fall short?
So basically, long story short, since the 60s, 70s, when we discovered L-DOPA, which is a precursor of dopamine. That's kind of still the main treatment of Parkinson's disease. So in Parkinson's disease, these neurons that die off in the substantia nigra, they're the ones that produce dopamine, and they regulate our movements. So because they die off, there's not enough dopamine, we can't control our movement, and we get the slowness, we get the tremors, and so on. So we substituted with L-DOPA. The brain takes it up, and it transfers, changes it to dopamine, so we can function normally. Fast forward all these years, decades, and not much else has happened. We've got some more additional treatments that will allow the L-DOPA, it's called Cinnamate in New Zealand, to work for longer, to have less side effects, and so on. So that's kind of like the main thing. The other thing that has come about is deep brain stimulation, which basically they put electrodes into your brain, and instead of the neurons sending out the signals, it's these electrodes that mimic the signals and replace it, which works amazingly well for a subgroup of people. Okay. Amazingly well. But again, it will only work up to a certain amount of years. And then, as we said, this cell death, these neurons die off, and they keep on dying off because we don't actually treat the underlying symptoms. There's a lot of investment in therapies. So every year there's this review of what's happening, and there's some things happening, but the majority is still focused on symptom treatment, not really underlying cause, because, as I mentioned, it's really hard to find the underlying causes. It can be different for everyone. So
the golden egg would be to figure out a way to break up the synuclein clumps, right? And no one's figured that out how to do that yet. That's still the...
Yeah, so there's been a few big studies using antibodies to detect the alpha synuclein and to try and remove the alpha synuclein. Didn't really work. They've now reanalyzed some of that data, and they saw that with the worst patient, the fastest progressing, there seems to be some response, but that was retrospective, so not sure if it works. The biggest problem with any of these therapies is that they start too late. So all of these therapies that we've tested, trialed in the past, you have to have a diagnosis of Parkinson's to be enrolled in the study. So as I mentioned, 70% die off. You only have a certain amount to play with. You can only improve so much. So if you can detect the disease earlier, you can start treating earlier. And that will make a big difference, because a lot of damage happens at the end, right? Yeah. So it's a very slow process. And then you kind of cross the threshold of no return almost, and then just damage accumulates, and it goes faster and faster.
I see. So how would one go about screening for those early signs of Parkinson's before they would get to that 70% die-off of the cells in the substantia nigra and thus qualify for a formal diagnosis? Are there risk factors? Are there genetic tests? Are there family history? Or are there other markers that one could use to say, oh, yep, you're on the track?
Yeah. So I could talk about this for an hour or two. Just to start with the easier one, there's genetic risk factors. We can screen for them really easily. We can do it the day you're born, basically. We can see if you have the mutation, yes or no. But we probably don't know all the risk factors yet. And the biggest problem is probably that about only 10 to 15% of people with Parkinson's disease have these clear genetic risk genes, that we call them. Yeah. So we're left with another 85%. There's a lot of risk factors, environmental risk factors. people with working in mines that are exposed to high amounts of metals are more at risk of Parkinson's disease because it dysregulates the metal contents within our brain. And this is linked very closely to one of the early symptoms of Parkinson's disease, which is loss of smell. So a lot of people, up to 95%, lose their sense of smell before they actually start getting the motor symptoms. Really important one. And another one that's been very highly thought now in Europe to prevent is the exposure to pesticides, herbicides. So paraquats has been linked quite heavily to causing Parkinson's disease. We haven't banned it yet because, as I said, between any of these causes and you actually showing symptoms, they can be as anything as a few days to decades in between. So we've got genetics, we've got metals, we've got pesticides, but viral and bacteria can also cause Parkinson's disease. Not necessarily immediately, but what it does, and this is why it's really important to potentially get vaccinated, is every time you get the flu, every time you get COVID, you get a massive inflammatory response. It's basically an insult to your body and brain. We kind of forget how much it impacts the brain. And even though we make a full recovery, in reality, there's something that lingers. There's a bit of damage that occurs. If you have multiple of these over a decade, it will basically cause a damage every time to the brain. And every time it's like a little bit more. So very gradually. So long story short of all these risk factors, how it was diagnosed in the past is you basically go to a neurologist, and the neurologist will look at you because in Parkinson's, there's a few typical symptoms. He or she will make you walk if you like have slow movements, or if you like hesitate, or if you have like that tremble. They will say, I think you have Parkinson's. We'll put you on Sinemet and see how it goes. And if it works, you have Parkinson's. If it doesn't work, you might have something else. It's kind of like, not all the case. In some cases, they'll do like an extra scan to exclude other stuff. But that's in the majority of the cases how Parkinson's is diagnosed. Last year, a massive breakthrough happened. And so these protein clumps of alpha synuclein, they're in almost everybody with Parkinson's disease. It's a very, despite the different Parkinson patients that there are, because the saying goes, if you've met one Parkinson patient, you've met one Parkinson patient, because every other Parkinson patient is different, different symptoms, and so on. They progress differently. So, but what's unique, what's communal with all of them is, of almost all of them, is alpha synuclein clumping. So what we can do now is we can take a piece of brain tissue or spinal fluid, and we can actually take that substance and we can amplify the alpha synuclein clumping. Because as I said before, once you have this clump, it can form more clumps. So we're taking this characteristic of the protein and its ability to clump, and we're using it to test it. So only in people that have Parkinson or that have the protein clumps available, we will see a positive result. And in people that don't have Parkinson's, we won't see it. And so that has been probably the biggest breakthrough in the last 10 to 20 years. And what is really the best thing about this is that of this, the whole list of people that they've analyzed, is a certain amount of them didn't have Parkinson's when they took the first spinal fluid, and they followed them up over a few years, and some developed Parkinson's 2-5 years later. And so they could see in some of them 2-5 years before that they have the protein clumps available. So you can now go back years before and see if somebody will get Parkinson's disease or get multiple system atrophy. And this will then have a massive impact because you don't have to wait until people get really sick.
Yes.
You can say, they've had this. There's like a 95% probability that they're going to develop the disease. We can put them on the treatments now. We can get them to do change their lifestyle now. And that will be, of all the things, that's probably the biggest thing that's happened.
Right. But to do a test like that, you would need a piece of brain tissue, which is quite invasive, obviously, or a sample of cerebral spinal fluid, which is less invasive, but still a little bit. Yeah, but
You still wouldn't want to do it
recreationally.
Yeah.
Yeah. But I suppose if you check the number of risk factors for Parkinson's disease, it would be a wise test to do, I guess. Would you say? Yeah.
Yeah, definitely. And so some of the research and what other groups throughout the world have done, if they've looked at see if you could do skin samples or if you could do nasal swabs or saliva, and there's promise that it works on those tissues as well. So if that's the case, you don't have to go, because nobody wants to purposely give up a piece of their brain. Spinal fluid is preferable, but even that is full of risk. So you don't want to do that if it's not done by a professional. So if you just a nasal swab, everybody's given nasal swabs now. It's almost like a second habit. So that would be the next step.
Okay. And then that could be just a part of everyone's routine annual checkup care almost, if it was that simple, little.
Yeah,
if
the GP will say, you have potentially constipation, you're sleeping badly, you're not smelling so well, let's just test for this.
Yeah.
It would be something.
That's really interesting. And so if that were, so that's future forward-thinking perspectives. Obviously, that's not happening right now. It's still very much the classical approach, whereas wait for the symptoms. The neurologist says, you've got this, put you on Cinemet, see how you go. But in that scenario, because, so if we have a test that can detect alpha-synuclein clumps earlier, but pre-symptomatic patients, there's still no clump-busting drugs out there. So the alternatives would be, is it lifestyle interventions? Is it just generally keeping healthy? Is it being active? What are these things that would be on the menu for treatment in that scenario?
So, and this is what I tell people, there's no magic pill. First of all, because Parkinson's disease is so diverse, not everything will work for everyone.
Yep.
But the classical things that work for heart disease work equally well for brain health. For instance, exercise is probably the number one thing to do for your brain, not just for your body, but for your brain. And you keep healthy. There's a clear link between being underweight and overweight to getting Parkinson's disease. So if you're normal weight, you're less likely. But the cool thing about exercise, and there's already a lot of studies that show that exercise works equally well and better against depression than standard medication. So there's no denying that one. But a few years ago, they found that during exercising, you produce this hormone called irisin. So an irisin is produced in your muscles. It can go into the brain. It's one of these hormones that can bypass the blood-brain barrier.
Right.
Because this blood-brain barrier exists for protect our brain, but it also stops medication, it stops viruses. But this irisin can go through the blood-brain barrier. And what they've shown, still in rodents, is that it can reduce the neuronal damage by up to 35%.
Wow.
So, and you can actually cheat the system. So what they've shown as well, so if they got the rodents to exercise, they recovered, but they also got some of the rodents that to take an irisin pill, and they showed a similar effect. So exercise, even though this irisin treatment is not available yet, we can make it ourselves, right? So exercise is probably the number one thing for brain health.
Yeah.
But we can continue healthy eating. Really important. There's a clear link between a lot of people lose their sense of smell, but a lot of people also are constipated before they get Parkinson's disease. It's a bit of a chicken and the egg question. Is it a consequence or a cause of Parkinson's disease? It's probably both. But foods, the longer it takes for food to go through your guts, the longer it has to cause damage, to cause inflammation, stuff like that. So it's logical that a good transit is really important. So food is really important. And one thing that is, people always love it when I say it, is coffee.
Yeah.
Because drinking coffee has been shown again and again to protect against Parkinson's disease.
Oh, there you go.
So the caffeine plays a main effect. So it's not just coffee, it's tea as well. So the caffeine plays a role in that it keeps you awake, keeps you more active, but it also keeps the dopamine in your body, in your brain for a bit longer. So there's like multiple aspects playing around. We don't know the fine detail, but we do know that there's a clear link in that as well.
Is there an upper limit? Because I drink a lot of coffee.
Yes, there is.
Okay.
Because it's about 2 to 3 cups a day.
Max?
Because the higher you go, you get into the risk factors of developing risk for heart.
High blood pressure. Okay.
Yeah. So you want to seek that middle balance.
Trade off. Okay, so 2 cups a day, pushing it if you go to 3. And there's also this concept of the gut-brain axis as well, right? Which is implicated in a lot of different things. And is that also part of this wider, Parkinson's disease discussion as well?
Yeah. So, and this comes back to the chicken and the egg.
Right.
So, in a certain group of people, we see these protein clumps within the nose for the first thing. So we see we can see them when they've died. We can look at the nose. We can see it in some people in the gut as well. And what they've done in quite a few studies is that they've given these alpha synuclein clumps in rodents into the gut, and you could see that it can then spread through the nerves into the brain.
Through the nerves, wow.
Yeah, because your neurons are basically everywhere in your brain. So once it's in a cell, it can be transported within, and it can then go between cells. So that's kind of like the whole thing, and why we focus so much on containing it.
Yes.
So it's fine that you have it, but if it escapes, then it's out. It's basically a bit what happened with COVID, right? We had a really good containment system. One person got through, and
Right. So is it the case that if like one little clump gets out, that will propagate more clumps in other parts of the body because they kind of reach a critical mass and they keep growing? I mean, am I sounding like an idiot? But
in some cases, it will. One would be enough.
Yeah.
In reality, because our body is so well equipped, one will probably be contained.
Yes.
Or one cell will die off and will be cleaned up. Another thing that we do is every night when we go to sleep, we dream. Most of us do, but we go through a stage of REM sleep. So in REM sleep, you're pretty much paralyzed, except for your eyes that move really rapidly. So in Parkinson's disease, that is disrupted. So one of the early symptoms there is that people with Parkinson's will accidentally hit their spouses because they're enacting their dreams and they're not paralyzed. So they're like heading out. So why is REM sleep so important? And why sleeping is so important is that during the night, your brain gets washed. So this whole system of your brain being washed, drained, and it also clears out these protein clumps. So even if you would have some, it might still be clear that way. So we can get rid of them at night. So if we have like bad sleep habits, jet lags continuously, it adds to it.
Yeah. Amazing. Why don't we pivot now to like the New Zealand context? Because I know that you're working with Te Titoki Mataora on a couple projects, which are one of which is very New Zealand centric, but the other one is a bit more technical, and we can talk about both. I saw in your background that you're working in one of your projects. You're working to develop a more New Zealand centric approach to the management and treatment of patients with Parkinson's disease, because there are many international bodies that support PD and PD patients and healthcare practitioners, but not all of those are fit for purpose in New Zealand. So what's the idea behind that project, and what's the ultimate goal that you're wanting to achieve there?
Yeah, so the backstory to all of this is I was told this story of a conference, a paraplegic conference, and it was a mixed conference. They're becoming more popular. So there was neurologists, scientists, patients, patient groups, caregivers, everyone was there. So the first day, all the scientists were talking and all the neurologists were talking about how they're going to cure the disease and how they're going to get people to walk again. And so at the last day of the conference, one of the patients talks, and he says, I don't want to walk again. I want to get my dignity back and not wet myself. Fix that and we'll get a long way. So that kind of hit me, because the scientists, they didn't mean any harm, of course, but they never asked the patients what they wanted, what their priorities were. And that's kind of like where this project with Te Titoki Mataora fits in, is it's kind of like talking to patients and seeing where the biggest needs are within New Zealand and how we can target those specifically. Because one of my biggest regrets, maybe not regrets, but like frustrations as a scientist, is that even if I would find a cure for a proportion of people with Parkinson's disease, it takes about 10 to 20 years from finding something to getting it into clinic. So it takes a long time. So what can we do today to help people? And that's kind of like the driving force of this project and of this interaction with different groups like clinicians, patients, and not just patients, because through the interactions, patient sounds very passive. It's like something you undergo.
Right.
So, and in Te Reo, there's a really good word, tangata whaiora. It's like person seeking help. So they're actively participating. So they're like taking an active role. And it's really also empowering, because one of the clear things that help against any disease and helps recovery is a positive mindset. And what we found through this conversation is that a lot of people, when they hear they have Parkinson's disease, it's like getting a death sentence. Even though it's not the case, and it can be really well managed, and you can live 20, 30, 40 years with the disease, just getting that news causes a lot of people to go through like a depression. Yeah. And a lot of people keep it to themselves. So we've identified now where the biggest bottleneck is. It's like these early diagnosed people, or the ones that know there's something wrong, but they're kind of doing the ostrich in the ground. They're like hiding the issues and pushing them further apart. So it's like trying to get to them to help them seek help.
Right, yeah. Because it's not a one-size-fits-all. It's how far you are along the trajectory, your age, I imagine probably gender and other factors have a play as well, and making sure that there is... So do I understand correctly that it's really creating a framework that all the different participants in the chain of care for these patients have the right tools in their toolkit, so to speak, at their disposal to help patients with Parkinson's disease.
Yeah. It's getting the right information to the right group, but also it's about giving just the right amount of information, because you can Google anything. And so if you Google Parkinson's disease, you can read for millennia and not be finished. There's too much information.
Yep.
And it's kind of like deafening and overwhelming to see all of that information. And the other thing that's really important is there's this dogma, and when you Google Parkinson's disease, you always see an old person. And I think there's still the classical image of that James Parkinson's drew in his original manuscript of somebody hunched over that we still use nowadays. So Parkinson's disease increases as we age, but there's a lot of people younger than 50, we call them early onset Parkinson's, that have Parkinson's. And it's not just that. There's people as early as 13 years that have Parkinson's disease.
Really?
So these are genetic forms, but again, it's like, it's not somebody that's 80, it's like somebody that's 30. So they're completely different. They have different needs, different support. They will undergo the disease quite differently than somebody who's 65 and so on. So it's getting that balance right.
Yeah. And you mentioned before we got rolling that it's about making sure that the product that's being developed, let's call it a product for now, is fit for purpose, is what the patients truly need. It's not whipped up, by committee, shall we say. You're getting active involvement by people out in the community or patients actively seeking care for their Parkinson's disease, right?
Yeah, it has to be something that they want to use, and that's not a burden. Because one typical thing is we can make an app like we do for a lot of things, but people with Parkinson, a lot of have tremors, so it's sometimes really hard to actually use an iPhone, for instance. So you need to take that into account. And people that are 80 might not be as tech savvy, whereas people that are 40, they don't care. They can do whatever with whatever device you throw at them. So it's finding that balance again and targeting the different groups.
Yeah. And so the ultimate goal here would be to have a really easy, simple to use toolkit, enough information for patients and healthcare providers and family to all have sufficient information to care for a patient really well within the New Zealand healthcare ecosystem. Is that accurate?
That's part of it. Yeah. And also to help people find help, because one of the I think it's something that's really important is the longer you keep it hidden and you're not seeking help, the worse it gets. So as I said before, exercise, eating has a tremendous impact, and not just in preventing Parkinson's disease. If you're diagnosed with it and you start changing your exercise routine, your eating, meditation has been shown really powerful as well, you can actually change the trajectory dramatically.
Right.
And it can go, some studies even show that exercise can have an effect within 6 months in how you change that trajectory.
Wow.
So it is massive. So, but if you then wait 3 years, you've pretty much lost that 3 years, whereas you could have like gained 5 more. So that's kind of like, Yeah.
I mean, that's probably a good prompting for me to get back on the exercise bandwagon too. It's easy to forget. It's like, I always find that I have all this information and understanding at my disposal. I know that I need to eat well, sleep well, exercise, but modern life just gets in the way, doesn't it? Flip.
It is true. It is true.
And you have, maybe in the last few minutes that we have, the other information, so the other project that you're working on with TTM has something to do with, you mentioned tears?
Yeah, so as I mentioned, we can detect these protein clumps in the brain. We're trying to see if we can do the same thing with tears, blood, and urine. And tears is something that we're specifically interested in.
Yeah.
Because Of all the biofluids, let's call it that way, tears, saliva, nasal swabs, blood, urine, tears are probably the purest, the least amount of contaminants compared to any other stuff. Because if you do a nasal swab, it's full of bacteria, full of, if you take your saliva, it's full of enzymes. Enzymes break down proteins, which is the one thing that we're trying to find. So it's still possible, but it's just harder. So that's why we're focusing on these other groups. And the same thing as what I've told before, we're trying to develop this because within a New Zealand context, it's really important to reduce the burden on healthcare. Specifically, for instance, for a neurologist to diagnose you. So in Auckland, it's about a 12-month wait to see a neurologist at the moment. So if you can bypass that through a simple test that doesn't need a clinician to get the spinal fluids, you're winning another 12 months there.
Yeah.
So that's the really important. And also, you can go rural and take somebody's tears, urine. It's a non-invasive test. We can go wherever. It's pretty stable, so we can do all of that within New Zealand, which is really important and powerful.
That's so. I mean, access and equity. It's kind of hitting all the buzzwords, shall we say, that are important. So it's like a lot less invasive. Don't have to do a cerebral spinal. Don't have to do a spinal tap. What's it called?
Spinal tap. Yeah.
Yeah, spinal tap. Don't have to get brain tissue, which no one would really want to do that anyway. You just need to get poked and prodded a few times to.
Oh, just once.
Just once. Okay. Yeah.
And you just put a little bit of absorbent tissue in your eye, 5 minutes, and that's it.
Fantastic. And you don't have to go into a hospital to do it, like a spinal tap would require, and you can do it before you would be showing symptoms, so you can get that early diagnosis or early prognosis and early intervention kind of roadmap.
Yep.
Really cool. Okay, so this is another project with TTM, and they've provided some support funding for that. Is that right?
Yeah. So TTM provide funding. They also provide us as network information, and the belief in this kind of study, because it all started with some seed funding for me to have a conversation with a group of people that I didn't know before.
Yep.
And these now are like a core group of clinicians. I work with Parkinson's New Zealand. I work with patients, a genetic counselor, like all physicians, physiotherapists, all a different group, all involved with Parkinson's disease, some of which didn't know from each other what they were doing. So it's getting everybody around the same table and just aligning everything. And that's really important and powerful.
Absolutely. I heard a quote the other day that said if a committee designed a horse, they would have got a camel. So it's like you wouldn't have. So it's important to get all the different stakeholders and players in a system involved in that dialogue. Yeah.
And to have everyone have like an equal say.
Yeah.
That's really important as well.
Awesome. Well, if people want to find out more about your work, your lab, and these projects, where can they go, Victor?
So they can reach out through my lab's website, dieriks.lab.com. So that's the easiest way to find me.
Awesome.
Yep.
All right. Well, thanks so much for your time. Really looking forward to seeing what comes out of these projects. I know it's going to be great. Do great things.
Okay. Thank you.
Thank you.